So Dr. Katherine Maniates, welcome to the show. And can you please introduce yourself before starting our conversation?
Well, thank you for having me back. It's nice to be here. So, hi, I'm Katherine Maniates. I'm a postdoctoral research fellow at Rutgers University currently. When I did the podcast, I had just finished up my PhD at Rutgers University, but my current research focuses on the genes that are involved in fertilization. So what are those proteins that we find on the surface of the egg that are required for the sperm and the egg to bind and fuse with one another? And so that's what I'm focusing on now. I'm also an NIH Erecta Fellow, which means that I do a combination of both research and teaching. So yeah, it's nice to be here.
Yeah, awesome. Thank you for being and having this conversation again. So I guess you touched a little bit on your changes in your position and the personal milestones that you have. So now you're doing research, but also doing a teaching research as well. So that is awesome. So before getting into the science too much, can we just talk about how did you got excited into science?
Yeah.
Share your journey, I guess.
So I actually come from a family of mostly historians. Everyone else in my family is very focused on the history and things like that. But I really enjoyed growing up, how did things work, these really technical aspects of things. I was lucky enough that my parents really fostered that, taking me to museums, so science museums and things like that. But really crystallized when I got to my freshman year of high school, and I took the freshman biology course, and we learned the structure of DNA. And that was the first time I experienced what the central dogma of DNA and genetics is, where DNA is transcribed into RNA, which is translated into proteins, and that just really felt like it made sense. So after that, I was great. I know that people who are interested in science, they go to medical school. So I went to college, and I was I'm gonna be pre-med, I'm gonna be biology major, pre-med. I got about halfway through college, I had done shadowing with physicians and stuff, and then I was yeah, this isn't right for me. It's not really something that I enjoy doing day in, day out, and I'm a little bit introverted, so it was really a lot of energy expenditure. But I had gotten the opportunity to do a little bit of research in a course, and that just took off for me. So I went ahead and I applied for undergraduate research positions. I was at a really small college, so they didn't have that many. So I went to Rutgers University in Milwaukee, spent a summer there doing research, came back, finished my undergrad, and actually went back there for my PhD, and then finished up there where I did my work on microRNAs, which was featured on the podcast, and now I'm a postdoc.
That's great.
Yeah.
Wow. Yeah, what a journey. And I think it's really cool that it highlights the early on experiences really guide us, through our career, and you get to really explore what you really like and love,
Absolutely.
So that's amazing. So as you're we can segue into the projects that you're currently working on.
Sure.
Yeah. So what research questions, areas that you're interested in right now?
Yeah, so as I alluded to a little bit earlier, I'm really interested on what are the genes that are on the egg surface that are required for fertilization. So fertilization has been studied for a really long period of time. It's something that people are fundamentally interested in, how do we end up having babies, where do they come from, what are the genes that are required for that. However, we know many of the genes on the sperm surface, but we know very few on the egg surface. And so there's this large disparity, which maybe isn't that unlikely with how research has been done in the past, where there's really been a focus on males. But we see this across pretty much every organism. So I've wanted to figure out what are more of those genes that are on the egg surface. So what I went ahead and did is I used the model C. elegans. C. elegans are these little 1 millimeter large roundworms, and they're awesome because they're transparent. So that means we can see fertilization happening live. And the other great thing about them is that they share about 60% of the same genes that you and I have. But as opposed to using humans, where it's not really ethical to do fertility research, these animals have a 3 to 4 day generation time. So this allows me to really look very quickly to see what genes are required. So I went ahead and looked for animals that had the sterile phenotype because they were not able to have fertility, and figured out what those mutations were. And from that, I identified this gene that we're calling EG7. These animals are sterile, but everything functions normally and looks normal. And this gene actually is required for glycosylation. And so that's when there's the sugar groups on the outside of the cells. You can think about it as like making the outside of cells sticky. And you can think about how that would be really important during fertilization, where you need the sperm and the egg to come together. So that's what I'm currently working on. I also have some other genes in my back pocket that I'll be working on when I eventually have my own lab to work with.
Wow, that's super interesting. So I don't know much about C. elegans egg. What's the structure of the egg shell that they have? Like, is there a jelly? Is it more like a thicker jelly, or is it more a shell?
It's not a shell. It's soft on the outside. It does have several layers to it, but it, unlike mammalian eggs, it doesn't have a zona pellucida, so that ZP layer. So you could think that the molecules required for fertilization probably are right there on the surface.
Wow, that's really cool. So do they, after they encounter with the sperm, do they get internalized? Do they disappear? What happens?
Yeah, the sperm actually it binds and it fuses with the egg to, just like in every other organism. So even though worms might not look like us, they have to do all of the same things. They have to bind the eggs, fuse with the eggs, start that process of really early embryonic development. The sperm has to move towards the egg. So even though these processes might look a little different in worms, they're pretty much the same across all organisms.
That's great. Yeah, that's the power of working with model organisms.
It really is. It means that I've been able to look at thousands of animals to be able to look at this research and figure out what are these mutations, how are they causing infertility? And I didn't mention, but the gene that I determined that was involved in fertility actually has a human homolog, phosphoglucomutase 3, which is implicated in many different diseases, everything from skeletal dysplasia to immune function, but it's never been studied in fertility. And so now I've got this model where I can study what's going on in fertility when we have mutations in this gene in C. elegans.
Cool. We have a few questions
from
the stream, so that's
Hello,
exciting.
everyone out there.
Yeah, so first question is, how close is a worm to me? So can you give some measures that we can actually feel the closeness?
Yeah, so they're actually pretty similar. So they have to do many of the same things that you and I do. So we typically, the off-the-cuff number we give is about 60% of the genes are similar. And the one that I often give as a similarity is like, we both have genes for insulin receptors, we both have to do that. Both have like genes for being able to move and taste and feel things. So there's a fair amount of similarity. Cool. So in fact, there have been human genes involved in fertilization that were found in C. elegans over a decade earlier.
Yeah. So I guess like the conserve genes that are essential for their vitality, like those have like higher homologies with humans. So although the overall homology might not be that high, the ones that are related to diseases are like super high. So yeah, I think that's a great question. The second question is, how does a scientist know that they are ready for a lab? This is a great question.
It's a great question, and I don't know that I'm ready for a lab, so I think that's it. I don't know, I'm going to wait until, I think it just happens at some point. You start applying for faculty positions, and you work hard, and hopefully it works out that you get a lab. I think our next speaker, who already has their own lab, will be able to speak to this a little bit better, so I'll look forward to that advice.
Cool. There's a follow-up question for the first one. Like, wow, 60% like me. Aren't they really tiny? And how do you work with them?
Oh yes, they are really tiny. They're about 1 millimeter large. So you can see them, they're like the size of the head of a pin. And the way that I work with them is I work with them underneath a microscope. So I'll be looking through with both eyes through the microscope. And then how I move them is I use this pick that is made of a piece of platinum wire and then a handle. And it's got a flat end on it. And the reason it's made of platinum wire is that you can heat it up and sterilize it, but it cools really quickly, so the worms don't feel the heat. And then you're able to move them from one petri dish where they live to another.
What do they eat?
They actually eat bacteria. They eat E. coli, not the dangerous one you find in your food. This is a non-dangerous variant, but they're able to eat bacteria. If you found them in the wild, they would actually be eating fruit that just is beginning to rot. So like the really ripe apples and bananas. Cool. You might find them in your compost pile.
Yeah, well, I don't want to know.
Some people don't want to think about that.
That's good. But aren't the eggs even smaller? This is another question. Like, how do you move those? This is so cool.
Yeah, okay, so the eggs are really small, and a lot of the time I actually don't have to move them because C. elegans are transparent, so I can see right through them on the microscope. So I can put them on a slide, and I can actually see ovulation occurring, the egg moving into the spermatheca where the sperm is kept, and see fertilization happening without having to do any dissection.
So you're doing everything in vivo, meaning that everything is happening inside the live body, and you're seeing through the animal to look at how the eggs are, and the sperm is doing their jazz.
Correct,
So,
yeah. So it's
that is
actually
amazing.
really good. But we can work with dissected eggs. We can dissect them out, and you can still see them underneath the microscope. They're not so small.
Cool.
Yeah.
Underneath it. They are small,
They're
but
small,
not
but
big
within
enough.
the limit of resolution.
Absolutely. Yeah, great. So how do you do the dissection that is so small? Like, do you do it with your hand? Do you have different devices?
So we actually use like needle tips. So you know when you go to the doctor's office and you get a shot, there's little tips on your needles, and you can use those needles to actually hold one in each hand, and you can dissect apart the animal.
That's
Yeah.
simple.
Mhm.
Great.
It's
So there's
really
a lot
niche.
of questions on the system, which is great.
Yeah.
This is great. So,
I always tell I have a lot of undergraduates who I work with, and many of them are interested in going to medical school, and I always tell them, you're gonna be really good at those like really micro skills.
Absolutely.
It's good for that very fine motor development.
if you can make a surgery on a worm, you can do it on a
Oh, you could definitely do it on a human then, so.
Great. Yeah. So yeah, we appreciate all the questions coming through the stream. So can you tell us about your position at Rutgers? Like, how does your typical day look like in New Jersey?
Yeah, okay, so I'm a postdoc. So let me first tell you what a postdoc is, because that's not something that we commonly think about or encounter. So that means I'm someone who's finished my PhD and is doing another level of training before I start my own research lab. I sometimes think of it as a similarity to medical residency. Like you go to medical school and then you get another layer of training after that. So my day-to-day looks like I spend most of my time in a research lab. So about 70 to 80% of my time doing that. And like a lot of positions, I do spend time answering emails in the morning, and I also spend time writing up my experimental findings to be able to publish them. So I typically start my morning off doing some of that, and then I'll start doing some actual experiments. So maybe an experiment will be I'll have to count the number of babies that a worm has on a given day. So I might do that for a few hours, and we have a really collegial atmosphere in my department, so we typically, obviously can't eat lunch at your desk if your desk is in the lab. That wouldn't be very biosafe. So we pretty much always gather for lunch, sit, chat. So sometimes we talk science, sometimes we talk life. And then in the afternoon, I typically do a little bit more research. Sometimes we'll have lab meetings where the whole lab comes together and we'll discuss. And then I try to leave by 6 o'clock-ish so that I can go and do other things that I like to do, like my hobbies and things like that. So that's a day where I would only spend in the research lab. I also teach a few weeks a year. And so those days I might split my time up a little bit more, where I'll do some research in the morning, and then in the afternoon I might go teach like a cell biology lab or something like that, and then see where I go from there. So I might split my time a little more those weeks.
And spend some time on grading, making those
Yes.
exams, like going through.
Prepping so that there are homework assignments, so that the animals are ready for doing in the cell biology lab. Yeah.
Very cool. Yeah. So it seems like So I guess the next question could be what do you teach? Let's
What do
touch
I
on that
teach?
question. Does it get in the way of research?
I think it's a very delicate balance. I really enjoy teaching. I think that it's one of the most rewarding parts of my job is to either work with undergraduates or graduate students in the lab or get to teach classes. I just teach undergraduates right now. Right now I'm teaching a genetic analysis, part of a genetic analysis course, so that's for genetics majors. And then the other course I'm teaching is a cell biology lab. And I actually do that under the tutelage of another professor up at William Patterson University in northern New Jersey. And so I just teach like one section of that, like a couple of weeks of that every year. Does it get in the way of research? I don't think so. I think that staying connected to the people who are learning and like who are actually the people who are the next generation of scientists is really an important part of my job. My job is not only to be in the lab, be the hands that are doing the research. There's a lot of parts of my job that could probably be automated, But what can't be automated is my brain and my training of people and sharing what the research is about and teaching those things. So it's a fine balance, but I think that I'm better in the research lab when I don't spend all of my time just heads down and I'm explaining what I'm doing, why we're doing it, and it really makes you consider what are those important things, what translates out of this, why do people care, should people care, things like that.
Yeah, totally. So yeah, not just like doing the work, like doing the part that assessing the work and also communicating with other people, like those are like definitely, you know, something that is essential that people outside of the science don't really think about that much, but actually it is a huge part of our job. So it's great to see that you're fulfilling those goals. So you talked a little bit about like going home and then doing something for fun. So what do you do for fun? Like what is your life outside of lab?
So I have really like tried to craft a life outside of lab that is enjoyable, because being a scientist can fill up as much time as you give it, but it's important to have activities outside of it. So a few of my main activities, one, I go to the gym like a lot of times a week because I like to lift heavy weights. So I go with one of my friends from work. The other thing is I'm in a book club that meets once a month. We read all sorts of different books, and it's how I've met some really good friends and moving to new places. And the last thing that I really like to do is bake. So I'm really on a big kick right now of making homemade bagels. So that's my other hobby. So oftentimes my lab will get to benefit because I'll make more than we can eat at my house.
I need you in.
I know you gotta come from California to New Jersey
to
buy coastal bagels.
Sounds amazing. So I've another question actually rolled in that I've heard it is very expensive to work with mice. So is that the same for worms? And what are the costs of being a scientist?
Okay. So it is expensive to work with mice. It is not as expensive to work with worms. For one thing, worms, like, you can keep a lot of them around for the same cost as you keep keep mice around. And I don't have to, like, if I'm not using them, I don't have to keep them in a place where we keep mice alive and going. They can actually go into our -80 freezer in a special solution, and then we can thaw them out 30 or 40 years later when we need them to do an experiment. So that makes it much cheaper to just be able to keep them if we don't have to keep them out and going. Plus, they live on these little tiny petri dishes, not big cages.
Yeah.
So yeah, it really like cuts down. So it's not free by any sense. And then the second question was something about expenses.
To become a scientist, like
Okay. So I would like to mention that becoming a scientist after my bachelor's degree, I didn't pay at all for my schooling. So I did pay a little bit for my bachelor's degree. I had mostly an academic scholarship. But then I was like, well, I'm not taking out any more loans. And I was lucky enough to have a college professor who told me, like, well, it's paid for to go to go for a graduate program in the sciences. You get paid a stipend for either doing research or teaching. So I got paid. It wasn't a ton of money, but it was enough to live off of. And then now I work as a scientist and get paid a salary. So yes, there are expenses to get through your bachelor's degree, but pretty much every graduate program, if you want to become a scientist, is fully funded.
Yeah, and a lot of scholarships out there, even they are not, might not look like they are fully funded, but there's a lot of scholarships to support, especially the graduate programs in STEM. So
Absolutely.
yeah, encouraging news. So there was a comment, just a fun one.
Okay.
Like, can you cryo-freeze me too? So,
We're not there yet.
Yeah. So yeah, it would be nice that if we can just like get frozen over
40 years later.
And,
I don't know, as the glaciers melt, I get a little nervous.
I know that, like, who knows what year that you're gonna wake up, right?
Yes.
So I guess if you can go back in time to 10 years ago, I don't know what stage that would be, but what
I
would
have just graduated college.
Oh yeah, great. So what would be the one advice you would tell yourself? So what would have been nice to know then than now?
Yeah, okay, I think I probably would have told myself to like relax a little bit. It's going to work out, like you'll put the work in. Like if your experiment doesn't work today, maybe it'll work tomorrow, maybe it won't work tomorrow and you'll find a new approach. But like what I do is important, but it's not saving anyone's life on a day-to-day basis. So it's okay if things don't work. and tell myself to like really enjoy those times of like being in your early 20s and getting to experience life and not just be so wrapped up in graduate school. So
and
have a better balance.
Yourself out.
Yeah.
Yeah, balance is always a challenge, I think. Okay, so back to a little bit of the research. So what is the most exciting result that you have ever gotten?
Oh, okay, I can talk about, this was a recent one, not that recent, over a year ago now, but I was doing a really big genetic screen where you look at a lot of mutants, and then you have to figure out what gene is mutated. And so I had done whole genome sequencing, and I thought I had figured it out, and I did an experiment that actually confirmed that result, and I realized my mutation is in this gene, and I didn't believe it. I like hyperventilated a little bit, went and redid the experiment over another week, and then I like broke down and cried in the lab because it was actually like I'd been working for months trying to figure out what this gene was, and I was like, this is a really interesting gene. We don't know that much about it, but we now know a new gene that's involved in fertility.
Okay, wow. What's the phenotype that you look for the screening?
Yeah, so I look for sterility. So if basically the worms can't have any progeny, any offspring. And so being able to then I go back to their siblings and see.
Tracking back on where the mutation actually occurred. How do you induce the mutations?
Okay, I use EMS, ethylmethanosulfate, which is mutagen. It works really well on invertebrates like worms and flies, not as well on vertebrates like us, which we're lucky, and it induces, at the rate I use it, a change in about 1 in every 1,000 base pairs.
Yeah, so you feed the worm with that chemical, and then they will induce these random mutations.
Yeah, I actually soak the worms in this mutagen, and then it's their sperm and eggs that are being produced that get mutated, so we'll see it in the next generation.
Gotcha. Okay, yeah. So, yeah, it's unfortunate when you are like looking for the target gene that you thought that had the lesion in them, and then like you find out later. So that's a mishap, but like still you were able to track down that,
yes.
And then you are now working with that more interesting gene.
Yeah, and it was I was really new in the lab at the time. Like it was like a year into starting my postdoc or so, and so this was like a new field because like I did my PhD in microRNAs, which are these small non-coding RNAs, and now I'm working in these protein coding genes, which are like a different type of gene, you know. So you're like thinking about like, do I really understand what I'm doing? So you're like double checking and triple checking everything. So it can feel very much like a good feeling, like relief when you figure something out.
The gene that you're studying right now, is that gene in humans too?
It is, yeah, phosphoglucomutase 3 that I talked about earlier. So it's in humans. It's involved in putting the sugar molecules on the outside of many of your cells.
Yeah, that's cool. So the sugar is not the sweet sugar here, right?
No.
So how does the sugar work in protein?
Well, you're asking like a very biochemical question there. So it can work in things like cell adhesion and cell fusion. It can also be an anchor on the outside of your cell. So you've got the membrane on the outside of your cell, and it can hold on to a protein. I always think about it like a balloon. You know, you've got the string on a balloon, and it can hold on to a protein that can do signaling on the outside.
Oh, so it's like almost like a decorate on the protein?
It is. So it's like certain amino acids or the building blocks of proteins will have sugar groups added to them.
And then they will have like different functions depending on what kind of sugars are coated on them.
Yes.
That is super cool.
Yeah.
So that's another level of, I guess, gene expression control.
It is, yes.
Awesome. So I guess what's the part of your job, I wanted to ask, like, what's the part of your job that is most rewarding to you and most challenging to you?
Okay. Most rewarding is definitely when I'm working with a student and they don't understand a concept, and then over time I just watch it click. A lot of times they don't even know that it's clicking, but then they're just like, X, Y, and Z thing, and I'm like, you got it. So that's really rewarding because they wouldn't have probably, like, they had to really think hard and work hard to figure it out, and it's so awesome to see them grow. The most challenging part of my job is when things look like they should work in an experiment or something like that, and they just don't work for whatever reason, and it's really hard to troubleshoot. And so sometimes that can feel really frustrating.
Mm-hmm. So you set up a hypothesis, and sometimes the results don't look
Yeah, well, it's not even that. Sometimes it'll be before that, like the technical things that won't work. Like you're using a protocol from someone else, and you realize like, oh, well, if they would have said this thing, phrased it this way, I would have done it this way.
Yeah.
So it's really important to ask all those questions if possible.
Really matter.
But sometimes, I don't know, just things don't work. Just like at your house, like you'll be like, that light switch worked yesterday, now it doesn't work today.
And trying to like, get all the conditions correct, so you can like
and
finally
control
do your
for it.
Yeah, control is so important.
Yeah.
Great. Yeah, as a wrap up, can you please share your future plans or goals, such as like new teachings or your job search or maybe some outreach projects?
Yeah, so I'm currently gonna stay at my current job probably for another year or two before looking for an academic job.
You recently got this really great grant.
Maybe.
Okay.
I won't, we'll see.
Yeah.
Hopefully I got a good grant. We'll just say that.
You shouldn't jinx it, yeah.
Yeah, I don't want to jinx it. And so look for a faculty job, and then with my outreach and stuff like that, I will probably be, one thing that I'm doing is continuing to revamp part of the cell biology class. That's part of my teaching, part of my fellowship. And then also just talking to people wherever possible and like telling them about what scientific research really encompasses.
Mm-hmm.
So,
yeah, amazing. So good luck with your future endeavors, and can't wait to talk more about that, like your projects, how they turn out, the results from your great the screen that you performed, and much more so in the future. But thank you so much.
Well, thank you so much for having me back, Jay. It's great to chat with you.
Absolutely. Thank you so much, Katherine.